Good afternoon, everyone. In the last class, we were discussing about several things. It's a kind of introduction we had. We discussed about initiation of replication. We also discussed about telomerase activity. End replication problem and telomerase activity. So in this class, we will revisit those topics, and then again we will add some more details, especially in the perspective of eukaryotic replication. So we know the model typically referred to as the replicon model, which is to imply that there is a replicator, a small sequence that is essential for any DNA to be replicated. You can think of something like this. Um, any DNA, any stable DNA with an origin of replication, or you can call it as with a replicator, can act as genetic material. Okay, that's a simple statement. You can take it for granted, or you can put it like this: any DNA without a replicator is not a genetic material or is not a genome. You can take plasmids, you can take viroids, or viruses, plant cells, plant genomes, animal genomes. Everything has an origin of replication, and there are corresponding set of proteins that are here generally called as initiators, which recognize the replicator and uh, will result in the formation of two DNA molecules. Okay, that's the general overview of replication. And then we also discussed about different types of replicators. Some here the number 9 indicates that there is a 9 base pair sequence. Same sequence is at least 4 times in E. coli, or 5 times, sorry. And then this is a 13 base pair repeat. You will have it several times like that. So in SV40, this is virus, and this is for yeast. They have different structures, and therefore, say for example, if you take a bacterial genome and put it into a yeast cell, say bacterial DNA you have taken and put it into yeast cell, it does not get replicated, because the proteins or the initiator proteins present in yeast are specific for yeast origin of replication. So there should be, within a given cell, if a DNA has to be replicated, there should be a initiator, there should be replicator, that is the sequence within the DNA, or you can call it as origin of replication, and its corresponding, not just any initiator proteins, just its corresponding initiator proteins. Okay. So we discussed this. There are 3 important proteins. If you can see, DNA A, DNA B, and DNA C. So these are the 3 proteins. When it is bound to DNA A in complex, when it is bound to ATP, it's in active state. It will bind to these 5 9-mer repeated sequences I showed you in the previous slide. And binding of DNA A with ATP will denature this region. Denaturing means breaking of hydrogen bonds. So you'll have these strands are separated here, onto which DNA B and DNA C. DNA B is the helicase. I hope you remember helicase is the one that breaks the hydrogen bonds further in using ATP. And then it comes along with the complex that is the DNA helicase loader. The loader is responsible for DNA helicase binding. So it binds to, helicase is bound around one of the strands like this, right? Okay, I couldn't show it the way it should be, but yeah. And then it moves in 5' to 3' direction. So the other strand is here, but helicase is only bound around one of the strands and keeps on breaking the hydrogen bonds that are there. It keeps breaking. So I hope you understood the role of DNA A. It is to denature this region. DNA B, and DNA B is the helicase. DNA C is to put the helicase around one of these strands. And then we have, then the helicases will break the hydrogen bonds. Then we have primase, which is the enzyme. It's a DNA-dependent RNA polymerase, or it is just an RNA polymerase, but it is specialized only to produce small fragments of RNA molecules, because DNA polymerase does not initiate replication by itself, right? It needs a 3' OH. So once that primase makes the primers, and then the DNA polymerases take over, or the holoenzyme comes in and gets arranged at the primers and then starts synthesizing. So this holoenzyme will come somewhere here, and this one will come to this place, two different replication forks. And replication of this one will happen in this direction, whereas the other one will keep on going the other direction. And I also hope you remember the parts of the DNA polymerase 3 holoenzyme. There is a sliding clamp, there is a sliding clamp loader, there are flexible proteins, tau proteins, and DNA polymerases. Okay. And then DNA replication keeps happening. I hope you remember leading strand and lagging strand. Leading strand is where DNA synthesis occurs continuously. In lagging strand, it happens in bits, because the direction of replication and the direction of polymerization are different. So it happens discontinuously, through formation of smaller fragments like these. Each of them is called as Okazaki fragments. And then we also discussed about the regulation of replication initiation in E. coli. And we discussed about what is methylation, what is semi-methylation, and what is hemimethylation. Here we don't have to discuss all this stuff, and you also don't have to remember this semi. It is just for illustration purpose. Hemimethylation implies that one of the strand is methylated and the other strand is not methylated. Whereas compared to this one, in both the strands, DNA is methylated. Where is it methylated and what methylates it? It is at GATC sequences. As shown here, GATC, you read in this way, or in the opposite one, it will read the same, isn't it? That's called a palindrome. So DAM methylase, there is a methylase called, the name is DAM methylase, and it is a sequence specific DNA methylase. It recognizes only GATCs and it methylates on the adenine. Where does it methylate? It methylates on the amino group that is present on the 6th carbon of adenine. So this DNA you're seeing here is methylated DNA, because both the strands show complete methylation wherever there is GATC, except in this initiation region. Then if origin of replication has initiated, then it forms this is because of semi-conservative replication, you have one older strand and one newly synthesized strand, this one, the orange one. There would be DAM methylase has already acted upon the parent molecule. So both the strands are methylated. But this, in the newly synthesized part of it, there is only it is hemimethylated. Why do we call that hemimethylated? Because only one side of the DNA, one strand of the DNA is methylated, the other one is new, and it's not yet acted upon by DAM methylase, so it is still unmethylated. So what happens is there is a protein called as SEC. SEC has affinity for GATC, methylated GATC in hemimethylated. Okay, it does not bind to completely methylated DNA. If this strand was also methylated, this protein SEC would not have been bound to this DNA. It only binds to, it has affinity for methylated strand in a hemimethylated DNA molecule. So SEC is bound here. If you think of it, if SEC is bound, then DAM methylase, the enzyme which methylates the DNA or the adenine in this GATC sequence, cannot interact with this DNA immediately. It is because this is already occupied by SEC proteins, right? The SEC proteins bind interaction with the replication or the site of replication, origin of replication is not very strong. So occasionally what may happen is that SEC might dissociate. Anytime there is a dissociation, then DAM methylase may recognize that place. Say, for example, in this location, you see here, SEC is there. If it dissociated, DAM methylase might come there and it will methylate the other strand as well. And once both the adenines are methylated on both the strands, SEC can no longer bind here. The same will occur, and eventually the whole DNA will become methylated, and then next round of replication can start. I hope you remember or you know that E. coli replicates at very fast rate provided good conditions. It just takes about 20 minutes to replicate, and the genome is too huge, right? So the replication should happen very faster. But at the same time, if it were not good conditions, then the replication should not happen if the conditions are bad. We discussed those things in the last class. Okay, I hope you remember this. For illustration purpose, or the way I want you to make notes, if you have to write an offline exam, for this sake, a quick notes would be something like this. You have methylated DNA. I'm trying to make a notes for it. And what is methylating it? It is because of DAM. I don't need to write methylase. I just DAM methylase will suffice my memory. And it will result in, say, methylation, and then initiation has happened. What does initiation do? It creates hemimethylated DNA. Every time you are learning any terms like hemimethylated DNA, I'm trying to spend a little more time so that you will try to remember. If I remember hemimethylated, I can write a story about it, right? When it is the case. If there is hemimethylated, then what is going to happen? There is a protein called SEC-A that would bind to it. SEC-A binding will prevent methylation, right? So because of SEC-A, there is methylation is, I can put it like this, DAM, like this. But occasionally, there could be a dissociation of SEC-A, and then there could be DAM methylase may act upon it, and then you will have methylated DNA. You can use either words or pictures for your memory. This is for quick look right before exam. Once you go into the exam for exam hall, you should be able to write the correct, accurate, or elaborate answer for the question. So in eukaryotes, the scenario is different. Okay, this is something new that we are discussing today. In prokaryotes, there is one circular DNA molecule, or maybe some of them have two circular molecules, and they have only one origin of replication. Okay, so one replication fork will work this way, and the other replication fork will work this way, eventually resulting in what is shown here, something like this. And topoisomerase will decatenate it and will result in two DNA molecules like this. It's simple and straightforward in time with eukaryotes. When there are linear DNA molecules, that's when, and linear, and they are actually very long, that's when the trouble comes. So in eukaryotes, there are multiple origin of replications, or you can call as something like this, autonomous replicating system. Okay, but origins is it works fine. So this linear why do we have multiple origin of replications? Because there are multiple DNA molecules, multiple chromosomes, We will read about chromosomes in the next lesson. I wanted you to have a fair understanding of this, so that we will be able to discuss cell division and other things along in the next class. Chromosomes, there are multiple, and there is one more, and they are very long. So if you had only one origin of replication, they will not be able to finish the replication on time. Okay, we will discuss about cell cycle as well a little in the next slides. So you need, because it's very long, you need to have multiple origin of replications so that you can finish it on time. So the problem would be, if you had multiple origin of replication, then you should have very tight control of them. Otherwise, it's going to be a little bit of trouble or discord within the replication. So here is an example about how, so take for example number 3 here. These are the 1, 2, 3, 4, 5. They are the origin of replications that are shown. And just schematically or just theoretically, you think of 3 and 5 have initiated replication, right? As soon as replication happened, you have, this is one origin of replication. Let me try again. Now you have, this is one origin of replication, and this one is another origin of replication. The same case is this here too. They should be immediately somehow repressed or prevented from reinitiation. As we will see in the next slides, how this will accomplish. And just think of it this way, that though they are inhibited, or they will not be able to reinitiate. And then you can think of 1, 2, and others also get activated, and then they eventually form 2. They will complete the replication like this. The point here is how replication, some of them may start early, some of them may start late, but then they will all cooperate. Once this one mixes here, that's the end of replication for that strand. That's how they will work together. And there should be a mechanism to prevent any reinitiation from the newly synthesized origin of replications. So here is a detailed mechanism. I wouldn't want you to mug up all this stuff, because they are details rather than just logic. But it's what, as a student, I think you should get used to some of these details. The first thing is, there is a replicator DNA, or you would call it as origin. And then we have ORC, which is called as origin recognition complex. That is something equivalent to initiator protein. Okay. Origin recognition complex, as the name suggests, is what recognizes the replicator DNA. That is this part. And then it will recruit several other proteins, CDC6. I hope you remember that something like CDC and CDK and so on, when you read cell cycle in cell cycle regulation in your cell biology. From CARP, there is a nice description of it. But we will only stick to the DNA part of here, but just let's see. So CDC6 will bind here to the origin of, ORC, origin recognition complex. And then it will be bound by MCM 227 and CDT1. This is the helicase. MCM is the helicase, and CDT1 is kind of helicase loader. Okay, they will come and bind to the origin of origin recognition complex. And then there will be the hydrolysis of ATP by CDC6, and then that will result in release of the loader. Loader is released, and CDC6 also is released now, and that is when the helicase, that is MCM, gets loaded onto the DNA. Right? It has encircled the DNA so that it will start dividing, or breaking the hydrogen bonds or denaturing the DNA. So in the slide, we have seen until See, MCM 2 to 7 is loaded onto DNA and nothing has happened yet. And then there are proteins called as CDKs. I hope you have heard those names as well. The entire description of these two, previous slide and this one, is from Saccharomyces cerevisiae. So there will be some, if you are talking about humans or any other organism, there will be differences. But yeast is the simplest eukaryote that we can, that is well understood. And these are things that will have more developments in the future. So just see what all you can remember. CDK is cyclin dependent kinase, right? You must have read that before. What is a kinase? Anytime you hear a kinase, it is an enzyme that will transfer a phosphate group, typically from from one protein to another protein or so. So kinase is going to, and this is also DDK, that is also a dependent kinase. DBF, some other protein is there. What they do is they get activated and then they phosphorylate the MCM. Okay, they will phosphorylate, and that is when they, because of the phosphorylation, you will have the association of several proteins, which are typically the most important ones is DNA polymerase epsilon. I think in the last class I also described about DNA polymerase switching, right? In that we have polymerase alpha with primase, and it comes and synthesizes a small RNA molecule, and then the classes are switched, that is, or polymerases are switched, and then epsilon takes over. In another case, delta takes over. So here I'll get back to this position where phosphorylation happens, and then some of these proteins, SID2, SID3, they are released out, and also DB, and the other proteins get associated with the helicase, and then we also have polymerase alpha and primase complex and DNA polymerase delta. They will come and bind and organize a replication fork. Okay, leading strand and lagging strand on both the sides will be synthesized, and replication fork will move on towards opposite directions. Here is something that I want you to remember. In the first thing, we will also look in the other slides. You remember the cell cycle, G1, S, that will result in G2, and then we have the either mitosis or meiosis. We will consider mitosis here, and then again G1, gap 1 and gap 2, right? And S is for synthesis. What is being synthesized? That's actually DNA synthesis. That is where DNA replication happens. All of DNA replication happens within S phase, and synthesis phase is for, DNA synthesis. And mitosis happens, or mitosis or meiosis, that's why we call it as M phase. We do not want DNA replication to happen in any other stage, right? It is going to cause a lot of problems. I think I skipped that particular picture from the textbook. You will see that if DNA replication happens anytime else, it is going to cause problems. Especially partially synthesized DNA should not occur during cell division. Then the daughter cells will not get proper genetic material and they would die out. Incomplete or extra DNA that might come, many a times it will result in inactive cells. So M phase, this gap junction, why do you call it as gap? Sorry, not gap junction, gap phase. Why do you call it gap phase? Right after mitosis, it needs to grow. It has to ensure the cell division, everything has happened, and it should grow to prepare for DNA synthesis. And afterwards, you have gap G2 phase, which is also a gap to ensure that all the DNA is synthesized and both these cells have both the DNA sets of chromosomes have synthesized properly, or DNA is replicated properly, and then prepare for mitosis. Okay. So what happens in G1 phase is there are the initiation of replication in eukaryotes is divided into two phases. One is making the helicase, loading the helicase onto the DNA is one thing. And the second part is activation of DNA, activation of the helicase. Here is something that I skipped in the ACE1 is helicase is loaded. It is two parts, they are bound together like this. But only upon activation of helicase, those two subunits will separate, and then they will start unwinding or denaturing the DNA. For that, you need certain proteins, CDC44 and others, which will phosphorylate and then will allow the movement of the DNA. DNA helicase. Yeah. So if you can remember or try to understand it, what you're seeing is that DNA helicase or the DNA initiation of replication in eukaryotes is divided. The key aspect is loading of helicase is one part, activation of helicase is in one part. I think you also remember that at different phases of there are checkpoints and during the cell cycle, and each of these checkpoints there are different cyclins and cyclin dependent kinases that activate different proteins. So in G phase, it will allow the loading of helicase, but the phosphorylation required for activation of helicase is not happening. So activation of helicase is not there. Whereas in SG2 and M, there is no loading of helicase, but activation of helicase will happen. Is there a problem that there is in G2 and M phase, helicase is still remains active? There is no problem because here you have the loading. I will try to use a different one. Here you have helicase loading, okay? And by the end of this, helicase would have finished replication, right? Even though helicase might be in active state, there is nothing to replicate. They have finished the replication. And only when new helicase loading happens, then second round of replication can happen. Okay, let's see in the figure that would be shown here. So G1, S phase, G2, and M phase. S phase is where all replication happens. In G1 phase, the CDK, cyclin dependent kinase level is low. So it will allow only the until helicase loading can happen. But because cyclin dependent kinases, which are required for activation of the helicase, is not there, they will remain inactive in this stage. Once in S phase, CDK levels are high. That means phosphorylation or activation of helicase will happen, and therefore the replication will be finished. Okay, so you can think of it like this. In G2 phase, origin recognition complex will bind to the to the replicator or origin, and but no helicase binding will happen. In G1 phase, you will have helicase binding, and only in S phase helicase will get activated, and that is required for replication to initiate. These might be complex or too many details, but you have to read it in one go. We might not be able to understand. So I would suggest that we repeatedly see it, watch an animation, read the textbook. That's the most important thing. And then you can watch these videos if you think you'll be fine with them. So the next part is what we have already discussed about end replication problem. I'm repeating these because I think they are very important logically, and it's also fun. Sometimes it might be complex to understand. What is end replication problem? Why does it arise? The problem is because of DNA polymerase. Why is it the problem of DNA polymerase? Because it needs a 3' OH. It cannot add, it cannot initiate replication without a free 3' OH. And if you need that, what is supplying it? It is being supplied by primase. Primase is making RNA, it's not making DNA, right? And when you are trying to replicate, we want everything to be in DNA. So at least at the ends, so the one that is shown in green here, in the ends, what happens is that this is the RNA molecule, and you will eventually assume that the replication fork has moved and these two are separated. And then what would you get? You will see that, you'll see that for the the leading strand, no issues. Replication is happening in this direction. It will replicate until the end. For the lagging strand, it is producing Okazaki fragments. The question will also be like, I will try to make a little bit of complexity if you can think of it. Let's see. I will erase that part. Oops, sorry. Okay. Actually, DNA will get, for the leading strand also, there will be a trouble. Why will there be a trouble? Say, I'm just putting half of it, okay? Don't assume that I'm doing the whole. So this will be the leading strand, right? So this will be 3' and 5'. Somewhere DNA replication has to start with a primer as shown here. And you have to delete that primer because that's not DNA. And then that gap should be filled. How will that gap be filled? It's difficult. The same thing also happens in this place where you are seeing that the green one shown here is the RNA primer. And here also there are RNA primers, but they are not a problem because once this part is removed or deleted, the 3' OH of this Okazaki fragment will allow the filling of that gap, and ligase will come and fill up the or close the neck. What about this prong? This end is a problem because there is no way of synthesizing it, right? Eventually, chromosomes were likely to become shorter and shorter after each round of replication. And if there are any genes, important genes here, and then cells will die. Telomerase is implicated, or telomeres are implicated in several diseases, something like cancer, for example. What do cancer cells do? They will keep on dividing. Okay, and this division will keep on happening. And if telomerase activity is not high, in each round of replication, what will happen? Chromosomes will become shorter and shorter and they would die. But in cancer, what happens is they usually the telomerase is highly expressed. That means they can actually divide continuously. The same is the case with stem cells also. Maybe you'll read about these in the later courses. So what does telomerase do? Telomerase is I find it really exciting because it has a way, we have seen the animation, and it's a kind of ingenious ribonucleoprotein. Why do we call it as a ribonucleoprotein? Because it has a RNA component and it also has a protein component. The RNA component acts as the template. So technically, telomerase is a RNA dependent DNA polymerase. It's making DNA, right? So it is a kind of reverse transcriptase. So we have a DNA molecule. It would have been something like this, the complementary strand, which would be having 5' and 3'. This end is what we are having trouble to synthesize. So telomerase doesn't do anything to this strand. What does it do? It does to the opposite one. It will synthesize, it will use this 3' OH and add the continuous bases here. And then the next step is called as translocation, which means that there will be the telomerase will move. So assume that AAU is now base paired with TTA here. After a while, if you see, it will move from here to the newly synthesized strand. It is coming to here. I mean, it is at the edge, and now this part will be synthesized. And eventually, it has the original strand was still here, and now it has synthesized till here. And this cycle will keep on continuing for a long time until a large amount of the strand is made. And how is that helping our purpose? It is based on this logic, right? This is the gap we need to fill. We are unable to fill it because DNA polymerase has a problem. So telomerase will extend this very long, onto which now a RNA primer can be made, and this part of the DNA can be synthesized, right? You are extending the template required to generate, onto which a RNA primer will be generated, and then the complementary strand will be synthesized. Okay, that's a very ingenious way of how nature solves problems. Um, the telomeres, they have a certain sequence, and I want you to also, before I move forward, I also want you to appreciate something here. Let's see. I'll give you a question, and I want you to solve it. I want you to find out something about this template and see which of the sequences I'm going to give. Assume that I have isolated a um, new telomerase, and I want you to find out which of the sequences is likely to act as a template in the telomerase. A, U. Just try to follow some logic, and that will help you to see. Telomerase has to translocate. So keep that in mind, and then you will be able to find out the answer. One second, sorry. How many of these sequences, you have to tell me the numbers, how many of these sequences can act as a template in a novel telomerase? Anybody? First thing is you have to, I will try to solve it myself. First thing is RNA telomerase is a ribonucleoprotein. So this is not an answer. Number 3 is not an answer. Why is that not an answer? I am using the process of elimination. Why is it not an answer? Anybody? 4 is also not an answer because RNA does not have thymine. Okay. Second thing is here you should have, we'll break this into 3 parts. In all of them, I just gave 9 bases you can think of. You can also see the same sequence here. Why should there be a certain architecture for telomerase? You can see it here. I will try to zoom it more. Right, I hope you see it now. AAU, and here is another AAU. Why should be that case? Right, it is required AAU, and when it is translocating, it should again base pair with another AAU, right? Okay, See, upon translocation, this should be able to come and base pair with this. If it has to base pair it should have produced TTA. For that to be produced, there should be a template here, AAU. I hope you understood the logic here, right? So if I have to break this part, it is in typically these are the important things. Being in direct repeats. Why do I call it direct repeats? I'm reading it AAU and there is AAU, right? If this was in, say, something else, something like I'll put, if I changed it to U, here it would have been A. Then this part of the template will not be able to bind to this region. Okay. I hope you understood the answer for that. If not, we can always discuss in the next class or now itself, if anybody has doubt. So I eliminated these two. They are not the answers because they have T, and telomerase is a ribonucleoprotein. It has a RNA template. Second thing is that this one is not in direct repeat. So it will not be useful. If this was the template, if this was the template, it will not be, translocation would not have happened in this step. The same goes with this. This is not a direct repeat. It's actually complementary. Here it is an inverted repeat. AAU, and this one it is AAU. That's not going to help. Here it is AUA, and this one is a complementary of this region. That's not going to help either. And the last one is what we have, ACU, then you have GGG, and then you have another direct repeat, ACU. Then translocation can happen. Okay, that's how telomerase template and its activity is determined, or that is how it acts. So these are some of the proteins that bind to the telomere regions. Telomeres are every chromosome, as we will see in the next class. They have three important parts. One of it is, the middle one is centromere, and the ends are called as the telomeres. Those are the ends of the DNA. Here, chromosome, when you say, it is bound with so many proteins. On the telomeres, we have certain proteins like RAP1 and so on, RIF and so on. This is in yeast. You don't have to memorize those. This is in humans. So they're just to, at least what we should know is there are several proteins that will bind to the telomeres. Why should they be bound there? What is the problem? Why do we need it? If there is a circular DNA, it is much more stable. There are no free ends. But if it is a linear DNA, there are free ends onto which exonucleases can act and start degrading this from ends. So these ends, or the telomeres, should be protected. How can they be protected? That is by binding of proteins like RAP1 and so on. Okay, the function of these proteins is to bind and protect the ends. That's not all. There is also the telomeres length is regulated, right? The more the telomerase is working and trying to extend the DNA or the telomere, and if more and more proteins are bound and it is within reach, then it will inhibit the activity of telomerase, and telomerase will will dissociate it, right? The more number of telomeres are present, more new telomerase binding these proteins are there, more inhibition will happen, and then telomerase will stop its activity, which means that after every cell cycle, if telomerase is not in sufficient quantity, telomere size will become shorter after each round of replication, and at the end, cells will no longer be able to survive. So typically, if not always, any differentiated cell has a lifespan. It's called as population doubling and so on. How many times a cell can divide further, like that, you'll see. So if telomerase activity is less, the number of times the cell can divide is also less. If the telomerase activity is high, then you will see that they can go for multiple rounds of replication or cell division. So stem cells and cancer cells are some things that have high telomerase activity, so they can divide for many number of times. The end of the replication, sorry, the telomere is not, it's still vulnerable to exonuclease activity. So it forms a something called as T loop by folding upon itself something like a structure like this. And there are also proteins that are bound, because of which the ends of the chromosomes are um, typically they are um, prevented from being degraded by the exonucleases. So there are viruses. In viruses, they have a different issue, right? In viruses, they have a different way of solving it. They have a protein called as it is called as priming protein. That is a protein. It has a tyrosine group. Tyrosine is a hydroxyl amino acid, so it has an OH group like this. It binds there, and it comes here. It binds somewhere around here, and there is a OH, and that is used by the DNA polymerase as primer, and that's why this is referred to as priming protein, a protein that is acting as a primer. You'll find some things really fantastic, especially in cases of viruses, ingenious ways of many things that have happened that are not present in other life forms. Here is one example. This is simplest solution, isn't it? Compared to production of primase, producing RNA primer, then removing it, and then filling up the gaps, and end replication problem, and so on. These viruses, they have a simple solution. Just have a protein called something like priming protein, which can initiate or act as a OH donor, onto which more bases are added and replication happens, right? This is the best solution so far. Okay, I think we have completed this lesson with origin of replication, sorry, the replication itself. There may be here and there some things extra in the textbook, but I think we have covered a great deal of details actually. So we have studied the chemistry, we have studied the enzymology, we have studied the mechanism, and then regulation, and we have finished also topoisomerase. If you remember, we have gone in steps like this. All the classes are typically overlapping like this, so that your understanding should be better. Please read the textbook, watch all the animations that are there. Any questions?